Hello everyone, Microbehunter here, and welcome to another weekly live stream. Today it's on a Sunday. Unfortunately, I had to move the live stream from yesterday until today because of an appointment conflict that I had. But I'm happy that you still found your way here, and I hope that you're going to like today's live stream again. Today I would like to put some glacial water, actually not so much the water, but some of the black stuff that I found on some snow about 2 weeks ago under the microscope. And I've got the sample over here, and you can already see in the video here that I'm collecting some of the top surface layer of the snow where I could find all of this black deposit, and I wanted to find out what it is, and basically here we are. First of all, as always, as every week, I would like to ask you if the sound is okay, because sometimes there are some issues, and you can also see that essentially I'm also keep on rearranging a little bit the layout of the video, so I moved myself now again a little bit into the lower right hand corner. I hope that everything's technically fine. People have already starting to comment here and are saying hello all across the world. So what I would like to do is like every week, if you have a specific question that you would like to ask, please put an @Microbehunter or an @Oliver in front of the comment. And then I'm able to find your questions much more easily, and then I can respond to them much faster. What are we going to do here today? Yes, you can already see in the back in the video here in the back. I visited the mountains about 2 weeks ago, 2.5 weeks ago, and even though it was actually quite a warm day, and it has been quite warm, interestingly enough, I could find several patches of snow there, some leftovers from winter, so it kind of survived a couple of months. And I simply collected a little bit of the snow, of the black material, and I've got it here, and I would like to put it under the microscope. I have to also give you a slight disclaimer here. Let me see where I can find the desk view, and the disclaimer is it's not going to be the most exciting sample. Okay, so you can see that, of course, obviously the snow already melted, but there's the black material is in here. I did, of course, already observe it. There are a couple of things that are recognized, pollen, for example, some plant material. We're going to look at it again. However, there will not be any microorganisms, or at least no interesting microorganisms visible. Bacteria, of course, always present, but they can be a little bit difficult to see here. So it's not going to be the most exciting sample. So just a short disclaimer, but I think if we want to explore our environment, then that's, I think the important thing is that we simply kind of try to satisfy our curiosity, and even if not everything is totally exciting. Maybe later on, for those of you who really wants to see some water specimens or water samples, I do have some water samples here on the side as well, so I can also put them under the microscope so that we can see some movement for those of you who are interested. As always, I also would like to, because I don't have so many specimens today, I would also like to take the time today to answer again a few questions that you might have. So even if the questions are a little bit off topic, then you can please, of course, also ask them, and I will try to take my time to answer them, okay? As long as they're microscopy related, it's going to be fine, okay? And I already see, I hope I don't forget anything here, I already see that there are a couple of questions, or there is already one question here. Can I clean eye lenses of my LabCam phone adapter with 70% ISO to remove small debris very visibly at 400x? So basically you want to use 70% isopropanol to remove small debris, eye lens of my LabCam phone adapter. Let's put it this way. You can, if you want, I don't know if the LabCam foam adapter should have. Look, I don't understand. For cleaning optics, you use alcohol to remove grease, and actually there should not be any grease or any oil or fat on the mobile phone adapter. So that's a little bit the thing. So I think it's a little bit of an overkill. Generally, alcohol can be used. However, always use those cotton swabs, Q-tips. Because they have a large surface area. Because if you use lens paper, you might end up scratching it. But so before using any solvent or so, use a brush to remove any loose dust, and or you can of course also use compressed air, but do not use compressed air from a basically can. So some companies sell compressed air in spray cans. These are used to remove dust from keyboards and so on. And you should not use those for optical surfaces because it contains some kind of a solvent as well. So generally alcohol only if there's really grease to be removed. Otherwise, I probably would not use alcohol because it's not necessary. Yeah. So let's do it like this here. Yeah, so you can see that I have already placed a drop with some of the black substance and material on here. And what I'm going to do next is I'm going to break apart the clumps a little bit. I've already tried this today in the morning, and I realized that it's actually much better if you do break apart some of the clumps here, because this way the cover glass is going to be much flatter on the microscope slide. And it's going to be a little bit easier also to see some of the specimens here. Again, please do not be overly disappointed. It's not the most exciting sample, okay? But still, okay. So let's put it like this. Yeah, and I think we're ready. We're ready to go. Okay. I'm always going to also have a short look at the and hoping that I'm not going to overlook anything. Yeah, so let's go over to the microscope view again. You have to turn up the lamp a little bit. You can see in the corner that I'm now using the 4x objective. Okay. And here we are. I need to focus a little bit here, and I'm going to use bright field first. Okay. And maybe as we scan through the slide, maybe we're able to see some structures or some things that are a little bit familiar here. Okay. I'm going to, again, another question. I'm going to try the pipette method to break up the dirt. Okay, yes. Yeah. These, by the way, these are disposable pipettes. Yeah, I bought a pack of 100. Another pipette that I have are those pipettes here, those microliter pipettes. You can also use those, but the tip is a little bit too narrow. So what I, in this case, suggest, if you want to use those, they're a little bit more convenient maybe to use, then I would actually take a scissor and I would cut off the tip to make it a little bit wider. Okay, so at low magnification, of course, we're not able to see a lot. Let me switch on the arrow as well here, so that so here we have a little arrow. Where's the arrow? Nope, I have to do that. So now I'm able to move the arrow around, and let's increase the magnification now a little bit. And as always, we have to focus. And I did find in the morning, textile fibers, synthetic fibers. So in other words, microplastics, which was a little bit disturbing, of course, but not unexpected. And as we go through the whole thing here, we're able to see a variety of unidentified structures. But something that we have here is, and maybe I'm going to zoom in a little bit here, is those long structures that you see here could be plant material, but I also have found some animal fur and animal hair. Okay, this could indeed be some animal fur or animal hair, and indeed, and what you can see here, as a matter of fact, as today in the morning I found the same one, and occasionally you do see some dots swimming around here. It's not so easily visible here, but if you look at the end here, then you see that the hair has a little bit of a different color, and I think this might be part of the hair root. So there were animals were basically in the area, so it shouldn't be surprising that you can also find some animal fur here as well. And sometimes if you actually magnify a little bit more, you can actually also see that the fur has a canal, is hollow in the middle, so that's also an indicator that we're actually looking at hair and not at plant material. Okay, so yeah, let's go through this here a little bit. Again, patience is always something that's necessary. And yeah, of course, lots of decomposing material, soil material probably, yeah, rain carried probably some material to the surface of the snow. Wind, of course, and I've seen today in the morning, at least in my other slide, quite a few, yeah, pieces of here it is, here we go. Okay, so maybe you can identify this here. Yeah, this here, I've seen also more of those. Maybe we go up with the magnification a little bit. What could this be? This is actually a little bit covered up, but very evident. I don't know this species, but this is a plant pollen. The pollen, a pollen grain. Look, there are some moving organisms as well. Okay, you've seen this here. Okay, yeah, but this here is most likely pollen, yeah, of a plant. I don't know which one, obviously, but it's nothing unusual to be found here. Of course, there are many. Many plants there in the area as well. For getting a better overview, let's start to go down with the magnification again a little bit here. Let me adjust the other filter as well. Yeah. I've also seen a little bit some fungi as well. This here, for example, could also be a pollen. I'm not up. I bumped into the stage. Now it's gone. Okay, let's give it another try. Yeah. So plenty of things to observe. You can do occasionally do a similar thing also with house dust, because house dust is also can be quite interesting. I've seen some interesting structures already, some unexpected structures also in house dust. Yeah. Yeah, lots of smaller debris. Something that did surprise me a little bit is because the sample has been now on my table for over 2 weeks. I did expect some microorganisms to reproduce, and basically I did expect more, yeah, more movement. Later on, I'm going to show you what you can do with concerning time-lapse videos. Yeah, so all of those fibers that you see could be, of course, plant fibers. Yeah. Usually some, yeah, decomposing material from plants. Yeah. Yeah, here again, this might be also again pollen grains. This triangular shape is a little bit indicative of that. Yeah. So here as well. Yeah. This here possibly as well. Yeah. So what I have, yeah, there are a couple of questions that I've gotten here. I sampled the moss from 5 or 6 different shady spots in different parts of our forest and kept it moist. It's been 10 days now, to be continued. Yep. What do you want, if you are interested in finding some tardigrades, moss piglets, then what I recommend is you don't have to keep it moist for 10 days, but the moss should be moist for a couple of days, and then what you do is you kind of try to rinse out whatever is in the moist, and the solid material that will then be washed out might contain also some tardigrades. I made some videos some time ago that if you use that the source of the moss is quite important. I've been much more successful in finding tardigrades and other interesting microscopic animals. I've been more successful in finding those in moss that has been found in the forest, because I suppose because it has not been dry for a very long time. Yeah, it was always a little bit yeah, lush and green, and I think this allows yeah, tardigrades and other small animals to survive better. Yeah. So far, I only have the usual species, Paramecia, rotifer, but no tardigrades so far. Should you look out for a certain type of moss? Yeah, as I just said, I do. It's not so much the type of moss that I found out, but essentially the moss that I used was quite green and lush. So sometimes the moss is a little bit dried up. and even though the tardigrades are able to survive also dry moss, I found more of them to be in fresh, lush green moss. And I think it could also be that because they like to cling to the moss, maybe there are tardigrades there, but maybe they simply didn't come off the moss. So you do have to kind of shake the moss a little bit in a little bit of water to try to wash them out. Yeah. Where are the microplastics? Well, I just I don't know if I'm able to find one here, but every now and then you do find synthetic textile fibers, and you notice that they're synthetic because they sometimes have a color which is different, blue for example, which you otherwise don't find very commonly in the environment. And then you know that they must have come from somewhere. And now I've not seen so many here right now. Yeah, okay, so this here is, let's zoom in a little bit here. This is definitely some, most likely some plant material or wood or something like that. What you're able to see here are of course the individual cells. Looks almost like in a longitudinal cross section here. Yeah. So it's also nothing quite uncommon. Yeah. So let's readjust. But again, a little bit surprising for me that there are not more. Moving cells here, because I would have expected that those that are present would have reproduced, or they're simply too small. Bacteria are certainly present, but maybe not in a sufficiently high concentration. Yeah. So let me see here. See, that's one of the examples when the particles are not sufficiently broken up. Again, up here on the top, some what appears to be some kind of pollen, plant pollen. Concerning microplastics, there are actually some laboratory protocols that can be used to extract them. I do want to work on that as well. Microplastics, unfortunately, are fairly common, and those that can be found, especially also in water samples, are not so much plastic pieces as such, but usually fibers, usually from synthetic clothing. You kind of washed out, and then they collect, and of course people have already also found those synthetic fibers and textiles in rainwater, for example. So let's have a look here. So I'm using now DIC illumination, and this DIC actually does change the colors a little bit. So what I'm going to do now is I'm going to flip out the filter, and it might now appear to be a little bit more boring, but the colors, but you are able to therefore see the colors in how they are naturally, and this sometimes also helps to find either photosynthetic organisms. So for example, I've found some algae today in the morning, on my other slide, because they are green, so with the green chloroplasts. This basically means that sometimes the natural colors are an advantage, because this way you're able to identify certain structures better. Overall, maybe the colorful appearance might appear nicer, subjectively nicer, but this can actually also be a disadvantage, as it covers up some of the more interesting specimens. So I have to always also play around a little bit with the condenser. Here we've been already. If you see something like this, then I recommend that you make a video with a time lapse, and occasionally you are able to then see movement better. Okay, and occasionally in the background maybe you can see this moving dot. There are some possibly bacteria or small ciliates or flagellates, but not in the concentration that I was kind of hoping to find them. Okay. So what I need to do, and that is kind of the next thing that I have to work on, I somehow have to find a technique or method how I'm able to purify certain structures here. So basically, how I'm able to separate maybe some of them, so that I'm able to remove all of the unnecessary parts. Yeah, so here that might be a little bit interesting again, like I've seen before, where I'm suspecting that this could be some fur. The reason why I suppose that, and I'm not able to see it quite well here, is because of the surface texture, and also sometimes I was able to see that there was a central canal, which is typical of fur and of hair. But again, I'm not entirely sure. But one of the things that it does not seem to be made of cells, because I'm not able to see the cross divisions here. And in one case, I've also seen that it actually started to become thinner and thinner and thinner. Here maybe a little bit. Look, like the one over here, which is an indicator that we're actually at the end. So it actually tapered off, just like hair or fur. It's also pretty long here. Yep, here it kind of tapered off. So I do suppose, I'm not entirely sure, that this is indeed possibly here. Yeah, but I'm always willing to learn new things, and maybe it's something else. Okay, so sort of unrelated, but can you do a series where you culture your own organs microbiome for medical experiments, sort of like the Thought Emporium? I don't know the Thought Emporium. I have heard of that. So basically, where I can culture the own microbiome. It is possible, and I have done this, of course, already before. It is possible to culture your own bacteria. However, there are, I'm a little bit hesitant of doing that, and not because I'm kind of worried about it for safety reasons, but I'm kind of worried that other people are going to copy that. And this can be culturing your own microorganisms, especially if they are from your own body, can be a little bit dangerous, okay, because we're really growing organisms, microorganisms, bacteria at a very high concentration. So for example, I'm a teacher, and we have certain guidelines of which experiments we are allowed to do and which ones we are not allowed to do using bacteria, for example. And culturing bacteria that are from your own body is not allowed. So we have to actually get safe bacteria from some E. coli bacteria from a laboratory or so, and that's fine, but not those that actually grow on our body for safety reasons. Viewing dust, would you mount it using water or Elmer's? Okay, that's a good one. You know this this glue. If you want to make mount dust, make a permanent mount, simply for observing the dust, of course, you just put it into water. Elmer's glue is for making a permanent mount, and it is also water-based. Generally, anytime when you use some water or water-based glues for making a mount, then of course the dust fibers or whatever you find in there will absorb the moisture, and some parts might start to swell. So for example, if you add if you've got dry pollen grains and they are much smaller, and if you add Elmer's glue or water, then they will start to absorb the water and swell. So essentially, any type of mounting medium or any medium that you add will also or might also change the sample around, simply because the mounting medium interacts with the specimen. So what would I use? Well, I would basically try both, or try different mounting media and see then which one actually works best for my purposes. And considering the fact that some of the specimens are not expensive anyway, you just might try it out. Usually when I look at for dust, let's say pollen, or maybe I want to find insect parts, quite fun, or butterfly scales, simply for observation, I simply put it directly on the microscope slide. I do not even add a mounting medium. I simply keep it dry to quickly scan it to see what's there. And if I want to get a better quality, then I add water. and if I want to then mount it, I'm not mounting dust so often, occasionally I am, then essentially I'm using some mounting medium. If you want to make permanent mounts of dust, then I recommend the following, because it can be quite interesting. Over the seasons, over the months, the dust composition changes. That you simply take a slide like this, put it somewhere on the top of your cupboard and allow the dust to settle. And then you basically can also this way make a determination of how many pollen, for example, are present in case you have allergies and so on. So this is kind of the, let me go down. Always use a lower magnification, and let's see if we're able to find some more. I was a little bit, every time you see different things, again, I spent quite a bit of time to observe this. I'm just going through the whole thing quite quickly. And what is this here? Could also be some fibers of plants, whatever. Here again, very clearly, that's one of the nice things about plants, because of their cell walls. They're so prominent, so that it's not basically difficult to see. So that's basically plant cell walls here. Let me quickly check here. House dust is hydrophobic, meaning it doesn't dissolve well in water. Maybe try vegetable. Yes, that's what I mean. You just try different substances, especially if they're, for example, depends really on which, because dust can be so different. If you've got a lot of synthetic fibers in there, it's quite hydrophobic, and then there are lots of bubbles if you add water. You may want to adjust the camera angle of the microscope lens over. This one here. Thank you for reminding me. Okay. It's a little bit. It's a little bit in a very unfortunate position here, because I might also. I've got the microscope controls right next to it. So let me. And I do have a possibility to focus it as well. I have to turn it manually. Okay, another question here. Thank you for your answer. Another additional question about the tardigrade search. Is it helpful if you divide the moss samples into smaller pieces and repeat the search after a while? Well, what I do is I do not. What I do is I don't divide the moss sample. I simply take the sample of moss, add some water, and I turn the moss upside down and just shake it out in the water, rinsing off whatever comes off. And then there will be a lot of particles in the water, and allow it to settle down. And then I take a pipette and I take some of the particles that are floating in the water and put it on a microscope slide. So I'm not really putting the moss itself directly under the microscope, but actually the water that was washed off from the moss. And I also found the following. If there are, this is also kind of interesting, usually if tardigrades are present, then you don't have to look very long. So there is no point in really looking for hours for tardigrade. Then just the moss might not be suitable. I've had some moss samples, I didn't find anything, right? And then I've had another moss sample once, and I found 5 or 6 of them just on one slide. So I think the bottleneck or the limiting factor or the thing is not necessarily only how long do you look and how patient are you, but maybe the actual moss sample itself. So I would simply collect different types of moss samples, and then try it out. So this is basically, and we can go on like this forever. Again, as I said, it's probably not one of the most exciting specimens, but I do have, because otherwise, you get the idea. And I have to tell you that again, today in the morning when I've done this, I saw more interesting things, like algae, some green material as well. I do want to show you now very briefly a time-lapse video that I made. No, that's the one. That's the wrong one. Unfortunately, just a second. That's not the one. Oh, okay. I might have forgotten to. That's a little bit unfortunate. No, That's okay. Here we go. Okay, so this is now 15 times faster. Okay, a time-lapse video, and you might see, yeah, not a lot of movement really. And this is indeed an indicator that there is not much movement going on, because usually there is a little bit of movement going on here in the corner, and I've basically zoomed in here. What you're able to see is the tiny particles moving back and forth, but that's Brownian motion. Yeah? And indeed, sometimes there are bacteria that are maybe connected to larger sand grains or so on. This causes the sand grain to move. Yeah? So, for example, this here is here top corner. But I've also made other time-lapse videos, yeah, or for example, there's a little bit of movement here. I've also made other time-lapse videos, and there was a lot of movement going on. Yeah, so I recommend that this that you use this a little bit as a check, yeah, to see if indeed there are yeah, moving organisms in there. And those particles that move here, that's most likely Brownian motion. So this specimen here is yeah, in that sense not very active. Okay, I do want to do the following. Yeah, another question: how do you distinguish between organisms moving and Brownian motion? When you see movement of organisms that are actually moving, like bacteria, so you definitely know that they're moving because they have a much more directional movement. Yeah, they're swimming across. Brownian motion is kind of, they stay more or less on the same space, but kind of vibrate back and forth. I mean, there are of course certain bacteria that yeah, also a little bit moving uncoordinated, but even then the movement is stronger. Yeah. And those particles are so small that essentially, yeah, even if they were bacteria, it could be that bacteria, they're moving by Brownian motion. But actual real movement is much more, in many cases, way more directed. Yeah, so it's almost like you know it when you see it. Yeah. I have multiple jars with pond water for my microscope channel, and some of them have some spoiled fruit with mold inside. They're full of microbes. Would that be dangerous? Okay, it's like this. You are growing unknown microorganisms. Okay, so that's number one. And for this reason, you should avoid direct contact with your skin. Actually, skin is a pretty good protection. However, you can never exclude the possibility that you have maybe some small injuries. So there is of course the risk of infection. And if you have basically moldy fruit or so on, don't inhale the spores. Some people are allergic. It's not healthy. Honestly, if you go, I don't know, maybe some of you have some kind of a compost pile where you put garden junk and cut leaves and so on. I mean, there is also a lot of decomposition going on there, right? It's also not healthy. So we have to be a little bit, how do you say, yeah, careful, but we should not become, how do you say, paranoid about the whole thing. Yeah. So generally, what I do not recommend is if you start growing, for example, organisms that actually are able to grow on decomposing meat, animal protein, because those bacteria essentially have maybe a higher possibility also to grow on human skin. Yeah. So this is a little bit the thing. Generally, I would always be careful, yeah, I would try to keep the samples small as possible. I wouldn't say that it is, I mean, I've got jars standing here next to myself as well. You have to observe proper hygienics. Yeah. So, and especially if you're allergic to inhaling spores and so, I would be careful. But I would say, yeah, what happens if you eat spoiled food? For example, if you leave some I don't know, some vegetables out there, some bread, and accidentally you eat it, it's also not healthy. So I would put it maybe into a similar category there. Yeah? So be careful by making sure that you always wipe off the things. So for example, when I go into a jar to pull out yeah, some sample, with those tweezers. What I do afterwards is I always clean those tweezers because I do not want to accidentally hurt myself. Yeah, because they're kind of pointed as well, right? And then you might have the risk of an infection. Yeah? So it's kind of a general precaution. Yeah, yeah. So okay. Let me see. I accidentally skipped. Okay, thank you. That adjustment was perfect. Okay, could you make a video about DIC, please? Okay, that is maybe yeah. That's a long topic as well. DIC, that is the technique that I'm using right now here. It's differential interference contrast microscopy. Yeah, it's I've had it planned, but maybe I'm going to make a video in my other channel for this because it's more technical in nature. So I hope that I've got everything here. Okay, so yeah, so that's basically what we've got here, and as I already mentioned, yeah, that's basically what we get. So I know that some of you were kind of hoping for some more exciting things. So I do have some jars there, but I do also have a second sample here, which I don't know really what to expect. This here is from a river, the sand of a river. And let me show here. And I said, okay, well, maybe why not also have a look at this here. And I'm going to put a little bit of the sand under the microscope, yeah, I don't know what to expect. Okay, so let's give it a try. So how am I going to get this out? I'm going to go in here. So the problem with the sand is, of course, that it's fairly thick. Okay, so this means it's kind of difficult to put a how do you call it, a cover glass on top. Okay, but we're gonna see. Okay, so maybe we have to work at a low magnification, but sometimes we are able to see some small, more or less small microorganisms between the sand grains. So this is, of course, not a very suitable specimen. So what we have to make sure is we have to kind of maybe knock it apart a little bit. It's way too big, so I need to remove some of the larger pieces here. Okay. And I might also want to add a little bit more water. Okay, let me use now the pipette. It's way too thick, obviously, so we have to use the low magnification alone. It's just enough to hold the cover glass a little bit. Okay, and let's give this one here a try. So, need to change microscope to the microscope view. Okay, and that's usually always the problem when there are too many. Okay. And let's close this a little bit. Yeah, so this is, where do we see that? Yeah, so this is kind of a little bit, it is too dry here. It's probably a sample that is better suited for stereo microscopy than for the compound microscope here, but that's better. Okay. Looks quite nice, I think. Yeah, but I have also seen this would be a typical case where one would make a time-lapse video, because sometimes you are able to see then the sand grains move around if other microorganisms start to bump into it. Sometimes they're even attached, and then I've made already some videos some time ago where there were some diatoms moving, and they were moving the much larger sand grains as well. Yeah. Yeah, so this is also, it's actually, I think this looks actually quite pretty. But let's go up with the magnification yet more. Yep. So, but usually, let's have a look. Yeah, also mostly sand grains, inorganic material, and because I'm using polarized light right now, of course they look quite nice and colorful. Yeah. So the point is I encourage you to yeah, simply experiment around a little bit here. Yeah. The reason why, I'll be very honest now, the reason why I collected this sand is that, oh, there's a little bit some movement going on here. Okay, do you see? Maybe you're able to see this tiny cell. This is definitely not Brownian motion. Yeah, this one was way yeah, too directed, but somewhere over here now it's not visible anymore. The reason why I collected the sand sample is because it's been said that there are actually small amounts of gold, even if you're in the sand. So I was actually hoping to find a little bit of gold here, but you'd have to separate the gold from the other material. I found actually something called fool's gold, which is actually, I think, some iron with sulfur. It has nothing to do with gold. Yeah, is in there. So that's why I've collected it, but. Of course, right now we're more interested in finding some microorganisms or anything else. But as you can see here, occasionally we are able to find some movement here. Yeah, see this one here? Yeah, this is not Brownian motion. This is yeah, already quite directed. Yeah. So, okay, you know what, I don't know, let me see, does the stereoscope work here? Ah yeah, let me try this just for the fun of it. Let me put this under my stereo microscope. So, need to focus a little bit. Yeah. Yeah, you get the idea. Yeah, it's way too, I think still way too dense. And what I'm trying to do in the future, still not found the time to do that. Last time I also showed it to you. I do have some microscope slides that have a small indentation here. They're a little bit concave, okay, and this can actually also be used as a place to put some slightly thicker samples, okay. But I think that even the sand grains might be a little bit too thick for those slides here. Yeah, but I'll see. Yeah, so that's kind of the next project that I have. Is to kind of try to do some kind of an extraction or purification process to kind of separate the larger sand grains from the other material that I've in here. One way of doing that would, of course, be by going to, according to density. So I could take the sand, I could then put it into a small tube, shake a little bit. The less dense material stays in the liquid. The sand grains, they will settle to the ground, and then I take the liquid on top and then I centrifuge it. This could also be used as a method to concentrate whatever is in there. Yeah, possibly some plant material, some organisms, and so on. Yeah, so that is essentially the the idea that that I'm going to be trying to work on. Microplastics I already mentioned. Microplastics can also be extracted using so-called salt water, because salt water is more dense, and if you add now salt water to let's say the sand sample or to another water sample, then the less dense microplastics will actually start to float to the surface. So there are actually some separation techniques for plastics as well, but of course it's not specific to plastics, and you then have to do another test, usually a staining test, where you can actually determine if certain substances are really microplastics or not. Okay, yeah, so that's kind of the a short thing. So far there are no more questions, so please do feel free to ask questions. Yeah, so what is another thing that I wanted to show you is I do have over here this sample over here. I'm just going to show you down here. This is a sample that I collected over a month ago from a pond. Some algae, some pond water, some you see that some sediment down here as well, and I simply left it open, standing open like this for a month now. Okay, and let's put a little bit of this under the microscope here as well, because I did find out that there is still quite a lot of life here. So I just thought, why not? So where are my tweezers? Okay, let me quickly clean this off a little bit. And I've also been looking for some worms here, because I did find some so-called Iolosoma worms. Use this here. And carefully separate this, whatever we have on here. The request for there's been always a request for me to make a DIC, to introduce the DIC more. The physics behind this is complex. I mean, I could show you some of the parts of a DIC microscope, but there is not so much to show you. That's a little bit the thing, unfortunately. So let's give this here a try. I think we do need a little more water here. And I've also taken out some of the worms here as well, because I wanted, and I've transferred them to a different container, because I want to actually see if they're reproducing. So I've been also feeding them a little bit. Okay, but let's have a look at this here, just for the fun of it. Okay. Always start with lowest magnification. Okay. Ah, here it is. I'm not happy. This here is Iolosoma. Oh, there's another one over there. Okay. So good. Yeah. That's you probably have watched one of my videos that I just recently uploaded about the asexual reproduction and of these worms, and about what it means to be an individuality. So these are very fun ones to observe. And what they do, a little bit blurry, is they are able to split in half over time, and then you've got two worms. They feed on microscopic debris and so on. On the head, if you zoom in, then you see those orange-red dots, which are oil globules. This one over here, this is again, I think this specimen is more interesting than the other one. This here, of course, is a so-called ostracod. Yeah, here is a smaller one. Yeah, the water crustaceans. Yeah, yeah. So there are comments here. Not an expert, but according to Microbehunter's advice, don't usually keep the jar. If you put the sample in a jar with a container closed to let algae get oxygen, maybe add food. Don't add food. Usually there is already plenty of food present. I always recommend to keep containers open because this allows more oxygen to go in, and then the larger microorganisms, ciliates, those worms, are able to keep the bacterial count low because they feed on the bacteria. But as soon as it goes anaerobic, as soon as there's not enough oxygen, then the bacteria will explode in numbers. Yeah, not so good. Okay, then the sample also doesn't look very nice. Yeah. For example, I give you, and we can actually observe this as well. Last time I also showed it to you. This jar here, I collected some algae and some water from the Baltic Sea. Okay, and I took it along in those plastic containers. I had to close them, of course, because otherwise I could not transport them. But already after 1 or 2 days, they started to smell very bad, and it started to decompose. And as soon as I was back home, I immediately opened it, poured it out, and after about 2 days, again the bad smell disappeared. Basically the other larger microorganisms started to feed on the bacteria. Okay, so I was just barely able to kind of save the specimen. Yeah. And so there are these worms here, and that's they're quite interesting to observe here as well. They are transparent. So under the stereo microscope, they're really difficult to see. They're kind of clear, white in color. What in the world? Look at this. Do you see this here? This might be the head, only the head or the tail of one of these worms ripped off. Maybe during the as I took it out and separated it. Yeah, so you see that it continues to live. If it's the head, then the chances are pretty good that it's going to grow a remaining tail. This one, I kind of decapitated this worm here, I think. Let's go up a little bit with the magnification here. But maybe that's also good because then we're able to, it's not able to move away as quickly. And then maybe we're able to see a few things better. So here we go. Yeah, I need to, it's now 40 times. Let's have a look at, okay, here we go. Yeah, and very typical of Iolosoma, if you see those dots here, these are oil globules, usually they're colored red. Maybe if I take away the DIC prism, yeah, we're able to see the color a little bit better. Yeah, occasionally we're able to, here they're not quite as pigmented, but sometimes they're quite red. Yes, it also maybe depends a little bit on the species that we have. Yeah, and this one is just the head of the worm. Yeah, ripped off. Yeah. So basically, I've torn it apart as I've taken the sample out. Yeah. And actually, it's I've never seen it that nicely before, because they're difficult to observe otherwise because they move around so quickly. Yeah. Do the oil globules on the Iolosoma serve as energy? Good question. Possibly. You see, there is the saying in biology, if you think it's conceivable, if you think that it can be seen as an energy storage, I mean, it's not totally out of the world, then probably it is, because in nature, nature is so diverse. Yeah, so. That you can find out pretty much anything. So if you think that they could be used as an energy storage, then probably it is. Yeah, because organisms have to be energy efficient. So if I can imagine that if they do not get enough food, that maybe they're going to feed on those oil globules. It's quite well possible. Yeah. Are these worms considered single-celled organisms? That's very clearly no. These are true animals. They are multicellular, they have cell differentiation. This basically means that they are made of different cells, and different cells are responsible for different tasks. So they have a very simple basic nervous system, nerve cells. So they are not single-celled. If you now put some larger single-celled organisms next to them, then you're going to interestingly see that they are sometimes not that much smaller. Okay, so for example, Paramecia and so on, they can be pretty large in comparison. Yeah? So this is size alone is not always a good indicator, interestingly. So for example, I've made some videos also about rotifers, which are also multicellular microanimals made of about 1,000 cells, and one paramecium is approximately the same size as one of those microanimals which is made of 1,000 cells. Yeah? Because Paramecia is simply so huge. Yeah? So again, that diversity that we see in the microscopic world is really large. Yeah? So, but let's go down a little bit again with the magnification here. So, but the question, of course, now is begs the question: is if these are actually small worms, microscopic worms, and you need a microscope. Per definition, they are actually not microorganisms, because the word microorganism kind of implies that they're single-celled. Yeah, it's a little bit of a question of definition. Yeah, yeah. Some people also call viruses microorganisms, but actually, strictly speaking, they're not organisms because they're not made of cells. Yeah, yeah. Again, our ostrocods here. Yeah. And and if you have those animals in a water sample, then the bacterial count will be lower because they feed on bacteria. Huh? Is it an X worm, or will it grow its tail back? Most likely it's going to grow its tail back. I think it depends a little bit on how much of the head is present, but those guys have a really crazy ability to regenerate. Yeah? So if you chop them in half, I've seen them actually grow back a tail over the course of a couple of days. But but the tail part, if you chop it in half, I've not seen grow back a head, but I've seen that the head part grows back a tail. Yeah? So again, this actually shows that I have no idea what this is. My first instinct would have been some kind of these tiny little feet here. I don't know. Yeah. So in that last worm species, are there males and females of the worm, or even most likely these are, as far as I know, hermaphrodites. They're both at the same time. And what they will do is they will also reproduce quite quickly asexually. This means they're gonna grow a new head in the middle of the body, and then they're going to split in half. Wow, this is some kind of this is an animal. Exoskeleton of some kind of an animal here. Yeah. If ostrocods are aquatic crustaceans, are beetles, flies, and butterflies terrestrial crustaceans? They are not crustaceans. Beetles, flies, and butterflies are insects. However, crustaceans and insects and spiders belong to the phylum called Arthropoda. So they are related, obviously, but they belong to the big category of arthropods, and among the arthropods you again have the crustaceans, the crabs and the ostrocods and the water fleas. Yeah, then you have the insects, and you also have spiders. Yeah, so and those tiny ostrocods, they belong to they're more related to the shrimp and the crabs, while the flies and butterflies and beetles and so on, they are insects. Yeah, I've not seen it here. For example, tiny mites. They are again more related to the spiders. What is this here? This here is a pretty short worm here. Yeah. So maybe this was also chopped in half. Yeah. And this here is definitely looks very much like some kind of a the exoskeleton of some kind of an insect. Yeah. Where is the best place to look for ostracods? Basically, if you've got a pond somewhere, a freshwater pond, you take some pond water and yeah, you just go for it. And I've, for example, collected, this is kind of interesting here as well. This one is, I think, over 2 months old already. Yeah? Some kind of a, it's been raining and there was water standing for a couple of weeks in a little, it wasn't a pond really, almost like a puddle. And there were plenty of water fleas and water crustaceans in there. Now, if you want to, if you don't have access to those and you still want to grow these things, I can highly recommend that you actually, especially also if you have children and if you want to have a little project with your kids, then over Amazon buy so-called sea monkeys, they're called. These are tiny shrimp eggs that you can buy. And then you can actually grow your own tiny little water crustaceans at home. And you can also put them under the microscope. I've also made videos of that. Yeah? But that is essentially essentially a more controlled way if you have no access to those. Yeah? Aren't they in the clade? Oh, pond crustacean could be possible. I don't know. Okay. So basically, there are in the systematics and in taxonomy, you can essentially there's this hierarchical level of actually categorizing those. Yes, when you have algae like these here, then you see those little round bubbles, and these are most likely oxygen bubbles. And yeah, if you observe them over time, you're going to see, of course, that they will grow because oxygen will accumulate. Yeah. Kind of, so this is really interesting in the sense that sometimes when you have a water sample and you observe the fresh water sample, you're going to see different organisms than maybe after a week or so, because there is usually some kind of a progression and some organisms will die out, others are going to be more successful, and here this one is evidently those ostracods are way more successful in this sample than others. Yeah, ostracods and those worms. Yeah. So plenty of worms. Yeah. There is also the possibility by looking at the type of organisms that you're able to find in a water sample, you can do an estimate of water quality. Certain organisms only like to live in certain water qualities, depending also on the oxygen concentration present, and some people are able to do some kind of a water quality assessment by observing the organisms that you're able to find. Yeah, so here, let's leave this here a little bit. Yeah, I've already received the request that I do talk a little bit about DIC. Just a second. Are there some more questions here? Did I overlook anything? Okay, I hope I didn't oversee anything. Yeah. What did I want to say just right now? Yeah, if some of you already requested that I talk a little bit more about microscopic techniques, I can do that, maybe about, I don't know, phase contrast, DIC, and so on. I'm currently planning out, because this was also a request to do an episode on so-called oblique illumination, where I'm using some oblique illumination filters. I need to get a different microscope for this, and I need to unpack one and get it set up, because the current one that I have right now does not allow me to put filters in, at least those that I want to put in. So I'd like to do this as well. So if you've got a microscope that has a filter holder, then this is something that you might then also want to try out. Some of you might have also already realized, if you're subscribing to the channel, I have started publishing YouTube Shorts recently, and this kind of pushed up the number of subscribers quite a bit. So YouTube Shorts seem to be quite popular. However, my long form videos, the regular videos, are currently suffering a little bit, because I have to tell you, I'm a little bit stuck myself, because I'm thinking on what type of videos I could make for you. Okay, and so if you have any suggestions of certain things that are interesting, that you're interested in, please do tell me. This channel here, I'm placing a primarily focus on observation, but if you want to have more technical videos on microscope optics or so, that's why I've got my second channel. So what else do we have here? Yeah. So you know what, just for the fun of it, I'm going to put in now, because I've also not looked at the sample now for 1 week, I would like to put in the one water sample from the Baltic Sea. YouTube wants to be like TikTok, yep. But I have to tell you the following, the viewing preference of people also changes over time. Sometimes people don't want to take so much time, and sometimes actually it's much faster and easier simply to look at a short video, 1 minute or 45-second video. But if for the longer videos, it's a little bit more, it takes more time obviously, and then if I'm taking too much time explaining things, and sometimes I explain too much, and then people kind of lose patience with me. So, but what I'm going to do now is I'm going to change the view again. So let's this one again is last time, a couple of days ago, there was much more white stuff floating on the surface. So it could be again that over the last couple of days the composition has significantly changed. So let's see what we have now. Okay, and this one here, for example, the pipette here tip, I'm not going to touch anymore, so I'm going to immediately trash this. Yeah, I've got a trash can over there, because I don't want to touch this here, because it contains a lot of bacteria. So I always. So let's have a look. If we're able to find here, where is the microscope view? Yep, last time it was still full with okay. Yeah, so basically not much happened since last week. I see pretty much the same. But I do have to increase the magnification a little bit. Yeah. Yep, so you see that there's plenty of stuff going on here. And if I want to see the natural colors, then yeah, and these are now the natural colors, and you see that there is of course also some green material also present, which are of course algae or possible cyanobacteria or whatever, which indicates again, yeah, lots of green stuff here, right? Yep. So this is a decomposing water sample. Okay. Because probably in nature otherwise from in the ocean you're not going to find such a high concentration of microbes, but because the water sample has been standing here now for a couple of days, the algae have acted as a food source, and this allowed other microbes to kind of reproduce and multiply. That's why we're getting these extremely high concentrations. Yeah. This here could be, looks a little bit like the shell of a diatom. Or not. Yeah. Yep, plenty of things going on here. And here, some of those larger ciliates. Ah, yes. This one is also something that I've seen. This is most likely, these are most likely cyanobacteria. Let me quickly. Yeah, it's a little bit. Yeah. This filament that's running across horizontally, it's gliding, is most likely cyanobacteria. They often appear in filaments, and what they do is yeah, they are single celled, but actually because they grow in these filaments, you can already say that it's a very basic form of multicellularity. They almost behave like one organism, but actually they're made of independent cells. Yeah. Yeah. So concerning the identification of the other cells that you see here, okay, these are not bacteria. These are yeah, most likely either flagellates or ciliates, single-celled ones. Bacteria are significantly smaller. That's number one, and number two, if you look carefully, you're able to see that in the cell there is something in there. There are certain cell organelles inside the cell, and this indicates that these are not bacteria, because in bacteria they are kind of homogeneous on the inside. They do not have individual cell organelles in there, but because we're seeing certain structures in here and because they're fairly large, yeah, these are protists. Okay, so again, a question for me. Have you considered getting an inverted scope? Any benefit over upright for such specimens? The inverted scopes are those microscopes that have microscope optics on the bottom. And inverted microscopes, you still look from the top, but essentially the lens, the optic is from the bottom. And the reason why you have those is because you're able to then put a Petri dish directly on the microscope. So you do not have to put it on a microscope slide, but you take the Petri dish directly on the microscope, and then you're able to look through the Petri dish from the bottom. And those inverted microscopes are very often used in cell culture. So when you're growing, for example, human cells in a Petri dish, then they can be observed. So that is primarily the advantage that they have. The thing is the following, I think, I've not tried it, but I think for very small specimens it might not be so suited, because the Petri dish itself is thicker than the cover glass. So essentially what you have to do is you have to look through the Petri dish, the bottom of the Petri dish, and therefore the distance is further away, and therefore you might not get the same high magnification that you're able to get with a high power optics that I have here, right? So I would say that it depends really what you want to observe, but I have to tell you, I also don't have any space here, and I would not be able to see things that otherwise would not be able to see. So essentially, for my purposes, I do not see any specific advantages right now. But if I were to observe certain organisms inside a petri dish, then this would of course be a different thing. So so here is again, here again, plenty of things that we're able to see here. It's quite remarkable, I think. Quite a bit going on here. Okay, folks, it's been 1 hour 14. So there are a couple more questions here. Can these get on me when I swim in the lake? Of course, honestly. I mean, let's put it this way: in a lake, the concentration will not be so big. One really has to be honest about this. This is a pretty bad-looking specimen. Okay, so that's why, I mean, you wouldn't go into a lake which kind of smells bad, and you also probably wouldn't go into a lake where you can actually see cyanobacteria or stuff floating on the surface. Then the concentration would be high indeed. But if it's if the lake is clear for swimming and okay for swimming because it's been tested, then of course there will be countless microorganisms present, but not at this concentration. So this is the concentration here is, I would say, almost unnaturally high. Obviously, because I'm kind of decomposing the material, right? These are essentially marine species here because it's from a marine water sample. What is this here? I don't know. Almost all of it looks like, I know it's not a pollen grain. So I mean, many people have asked me, oh my gosh, when I go into a pond, are they also going to be there? Well, of course, because it's part of our natural environment. Especially, for example, if you try to scratch some material, if you step on a stone and it's very slimy and slippery, well, then that's the biofilm where you have microorganisms growing there. But that's not a problem. That's why I'm saying they are not growing on our body. And that's why it is actually a problem if you were to grow microorganisms that are able to grow on our body. That's something I would avoid. This long thing here is a diatom again. Yep. Yeah, okay. Basically I started off this session by putting a specimen from glacial snow under the microscope. I ended up somehow showing you a couple of worms, some sand and some other things here. So let's put again the glacial snow under the microscope here. And yeah, in contrast, and you see that there is a huge difference in what you're able to find. So I think it's also one of the interesting things about microscopy that it's almost a little bit like chasing down things that you want to find, and then if you find them, tardigrades, whatever, and then you're all happy that you found them. But sometimes, things are not quite as as interesting. But actually, that's not correct. Even specimens like this can be interesting if you try to find out what the things actually are that you're able to see. So, for example, here, as I mentioned before, I was able to find a lot of pollen, for example, which are otherwise not able to see. So, you know what? I'm going to leave it at that. It's 1 hour 18 minutes already. Those microscopy sessions usually I always like to do in a way where even sometimes I myself don't fully know what to expect. But sometimes indeed there are some interesting observations. In other cases, yeah, there is less going on, but still, this still shows us something about our environment. Yeah? Okay. Yeah, so what I'm going to do now, people, is I'm going to leave it for today. I do plan, obviously, on doing the live streams every Saturday. For every now and then, I do have to change the schedule a little bit. If you're not sure about something, then do check, please, the community posts in my channel. I will always put announcements there if I have to move the live stream. Okay. Otherwise, yeah, I think I'm just going to say bye-bye for today again, and yeah, I'm going to leave it at that. I hope that these videos and live streams kind of also motivate you a little bit to yeah, keep up with microscopy a little bit, because sometimes yeah, people need motivation. Sometimes I need motivation as well. Yeah, to kind of keep on rediscovering our environment a little bit. Okay, but I'm going to leave it at that for right now. I'm just reading the last comments here right now. Yeah, but I think it's fine. There's also usually a little bit of a couple of minutes time delay from when I say something until it reaches you. But I think that's going to be it. Okay, I wish you all the best. Okay, see you around next time. Check out the YouTube shorts that I'm publishing right now, and some videos if you have any specific things that you're interested in that you want me to look at under the microscope. Please do comment. Otherwise, that's it for today. Happy microbe hunting, and bye bye. See you next week.